This is a video presentation on transcription in prokaryote. In the previous video, we discussed about the basic requirements about transcription, which are the RNA polymerase enzyme, the promoter site, and some other basic discussion on or an outline about the transcription in general. So in this presentation, we will have a discussion on transcription in prokaryotic system in particular. So these are the three important elements we need to know before moving into the discussion. So the polymerase enzyme, the major enzyme involved in the synthesis of RNA molecule. The promoter site is the region which is demarcated in the gene for recognition of the or recognition by the polymerase enzyme, and the transcription factors are the proteins which are involved in the helping or supporting the interaction of the polymerase enzyme precisely to the promoter site. So these are the three most important elements which we need to understand. So it is simply like suppose we have the DNA molecule. Suppose this is the DNA molecule, and this is the region what we denote like +1. From there the gene starts, and so definitely immediately upstream to the +1 site you can mark certain regions called the promoter. So this is the promoter. So this is the promoter region. So in the promoter region, we know that there are different types of promoters and different sequences are there. I'm not going into that. So we have the polymerase enzyme also. So we got the polymerase enzyme. Suppose there are different subunits and all those things are there. This is the polymerase enzyme, and I am also. So this polymerase enzyme is there in the system, as already we know that in E. coli, like in a bacterial system like E. coli, there will be many number of polymerase present in the system, and this polymerase has to come and interact with this promoter region. So this interaction is facilitated by another protein called transcription factor, and the transcription factor is a protein, an external protein, which direct the interaction of the polymerase enzyme precisely to the promoter site. So the promoter site of a gene is being recognized by the polymerase enzyme with the help of the sigma factor. So the sigma factor along with the polymerase, the sigma factor and the polymerase enzyme will identify the promoter region, and this actually causes initiation of transcription. So these are the three elements. That means we have the polymerase enzyme, we have the promoter region, and also we have the transcription factor. So these three elements are important for the discussion of transcription in prokaryotes. So this details already we know about the structural features, the different subunits of the RNA polymerase enzyme. These are the two largest subunits. Usually they are associated with the opening of the helix and all, and the alpha subunit is the most important thing you have to note is the function of the carboxy terminal domain of the alpha helix, which is helping the enzyme to interact with the promoter site, and also the omega, which is in terms of the function which is least important. And about the promoter, bacterial promoter also, like we have different sequences at the bacterial promoter site. They are designated like at -10 we have one at -35, and we have additional sequences like up elements, and still we can also list many such sequences, but these are the most important sequences, and of which the -10 and -35 are very essential, and the sequences are also given there like TA, TA, AT at -10, which is also called the Pribno box. right? All the additional sequences in the promoter region will increase the frequency of the polymer or frequency of transcription of that particular gene. So such promoters with additional elements are commonly, conventionally we can say that these are the strong promoters. And here comes certain basic features, that there is no primer required. It can start de novo with the addition of the first nucleotide into the system, and require all the basic elements. That means all the NTPs are required. Then the synthesis is usual in the 5' 3' direction of the RNA molecule, and of course it will be in the that when you consider the template will be lying in the 3' to 5' direction. And the elongation is pretty high speed mechanism, that around 50 to 90 nucleotides will be added per second. And this is another one, that is there is no proofreading like 3', 5' proofreading or 5', 3' proofreading, which is unique in DNA polymerase enzyme during replication, but such a domain is absent in RNA polymerase. But you can see certain references, like there is a kind of correction mechanism called backtracking in RNA polymerase, that we will see later. So when we see the prokaryotic transcription, for understanding we can split the entire mechanism into 3 phases: the initiation, elongation and termination. So the initiation is nothing but the interaction of the polymerase enzyme into the DNA, or specifically into the promoter site. So this occurs usually, as we know that the RNA polymerase is comparatively a larger molecule, so which will come and occupy at the promoter site, means which will cover an area of the DNA. So which usually binds at around 70 base pair upstream to around 30 base pair downstream, and that causes formation of a complex called closed complex. So this is the DNA molecule. So this DNA molecule will interact with the promoter region in the spanning the downstream and upstream, and the most important thing, which has to cover the start site. That's very essential. And the interaction is when we analyze in detail, you know that the sigma factor is important in the interaction. The upper part of the image shows the sigma factor consists of 4 different domains, and each domain will interact with specific regions in the promoter sequences. So there are 4 domains, and each of the domain will interact specifically with specific sequences. Like you can highlight the sequences like -10, -35, and you can see there this domain 2 and domain 4 are interacting with the -35 and -10. The other regions will interact with some other promoter sequences which are not shown in that previous image. And also you can see the other interaction of the alpha CTD, that means the carboxy terminal domain of the alpha helix, which interact with the up element, right? So this is the binding of the polymerase enzyme to the promoter complex. So this is what we call the closed complex. Now this closed complex, after the successful attachment of the polymerase enzyme into the promoter site, will cause, or there is a transformation, or there is a transition from the closed complex into an open complex, in which actually there is a melting of the duplex. That means melting of the DNA double strand at the promoter region, covering the +1 site, and usually that the distance or the number of bases melted is usually some around 18 base pairs. So this process is called melting of the duplex DNA. This is done by the polymerase enzyme to form an open complex. So now the closed complex is converted into an open complex. So this will be the story, the entire story, like the polymerase is there, which is you can resemble it like the palm of a hand. So inside the palm the DNA is there. DNA molecule is being that means the open site you can see, and there is a channel called DNTP channel, and through which the new NTPs, that means the substrates, will come in this region, and the newly synthesized RNA molecule will pass through the other channel. So this will be the entire story. But this is not that simple as we think, because for the initiation, suppose if there is a synthesis of the initial segment of RNA, that means at around 9 to 12 nucleotides are being added into the template DNA, then the polymerase enzyme now has to move further. That means the polymerase has to displace in the direction of synthesis. But for the displacement, some energy is required, first thing. The second thing, there should not be any blocking materials. So when we closely analyze, there is no energy for the system, first thing. The second thing, there is a blocking element, what we call the sigma factor here. So the sigma factor will not allow the easy movement of the polymerase enzyme along the DNA duplex. And the first thing which I noted is there is no stored energy for the system. So the energy has to be acquired, and this is being acquired from the system and even within this complex itself. So for that there is a mechanism called, or during the initial phase what we call, there is an abortive initiation usually. So in which the synthesis occurs in an incomplete manner. That means there will be addition of around 9 to 12 nucleotides, but that will not be elongated further. Right. So once the sigma factor is removed, then which causes the free RNA polymerase to move further. So there will be synthesis after the sigma factor has been removed from the complex. And that process what we denote is like it is called the promoter escape. So the promoter escape means the movement of the polymerase enzyme from the start site along with the synthesis of the new RNA molecule in correspondence with the template strand. Now the enzyme or the complex shift from the initiation to elongation stage. So the elongation is a normal process as seen in the replication, or even it's similar to that in eukaryotic elongation. It is simply the addition of nucleotides into the 3' OH of the newly synthesizing RNA molecule. The main difference in this thing is that we know in case of bacterial mechanism, suppose this is the site where the transcription occurs. So this is the region, and we have the RNA polymerase enzyme. The RNA polymerase enzyme is bound over here, and once the sigma is released, the enzyme can move in this direction. So here the newly synthesized RNA molecule, suppose this is the +1 site, from there it is going to synthesize. So the first base will be added. So after the addition of the first base addition is a unique feature of the RNA polymerase enzyme. That means the first base, it is like suppose this is the first base, this is the sugar molecule of the first base, and there will be the 3-phosphate group at the 5' position, and there will be the OH group at the 3' position. So this molecule will now interact with the template strand at the +1 site. Then now the polymerase can do the elongation. Like in the initiation itself, there will be addition of nucleotides here. So around 9 to 12 nucleotides will be added. So this is what we call the DNA RNA hybrid. So this hybrid has to be elongated further during elongation. But the elongation, as already noted, if the sigma factor is present at this region, actually this is the channel where through which the polymerase has to move, or the DNA molecule will move back. So once the sigma factor is released from the structure, this enzyme can move. So before that mechanism, what happens is this synthesized RNA molecule will abort. So a number of RNA molecules will be released from this structure. So that's what we call the abortive initiation. So after the abortive initiation, when the sigma is released from the system, the polymerase will move. The polymerase will move in this direction. And definitely there will be synthesis of new nucleotides. At the same time, what happens is the newly synthesized RNA will come down from the complex. So this will be the mechanism of elongation. So the elongation occurs in this region of the gene, and there will be the synthesis of new RNA molecule. That's what we call the primary transcript. So there is elongation process going on, and this continues until the system or the transcription machinery encounters a termination signal in the gene. Yes, then transcription elongation complex. This elongation complex is nothing but the complex of the DNA template, the growing RNA transcript, then the RNA polymerase enzyme all together, and also at the site where the transcription actively happens, there will be the melted DNA duplex, and that region will appear like a bubble, and hence it is called a transcription bubble. There will be a bulging of the duplex. Hence it is called a transcription bubble. And all the other things we already discussed. And about the energy requirement, the incoming nucleotides and the pyrophosphates which are released undergo a cleavage in order to supply the energy for the elongation process. And in the beginning we said there is no proofreading for the polymerase enzyme, but still you can see a correction mechanism in bacterial RNA polymerase enzyme, and this correction is done by the same enzyme's active site. That means the RNA polymerase active site itself will do the correction process also. So the correction is what we technically called backtracking. So backtracking is nothing but there will be a pausing of the RNA polymerase if the added nucleotide is a wrong one, and the wrongly added nucleotide has to be removed from the system, and then the synthesis will resume further. Fine. So for that, normally this happens as an intrinsic process of the polymerase enzyme, but the reaction will be enhanced or facilitated by two factors called GREA and GREB, and they are usually involved in the correction process. Okay, fine. Now we are moving into the third part called transcription termination. So the termination in prokaryote is comparatively well described. There are two types of termination mechanisms explained. One is called row dependent process and the other one is row independent process. And intrinsically to the when we analyze the genes will have certain sequences which is helping the termination, what we referred as terminator sequences or terminators. Right. So in the row dependent process, as the name indicates, there is a protein, external protein involved in the process called raw protein, and with the help of the raw protein the termination will happen. And in the second one there is no external protein, and hence it is, you can consider this as an intrinsic mechanism, and both the type of termination process occurs in different genes. And we will first see the row independent mechanism. So for that, what we have to understand first is the sequence at the terminator region. So at the terminator region we have at the terminator region we can see a particular sequence which is what we referred as an inverted repeat. It's also otherwise called a palindrome sequence, and which is followed by an AT rich region in the DNA molecule. And you can see a little more clear here. So there is an inverted repeat and followed by the AT rich region. So once this region is being transcribed by the RNA polymerase, they'll produce a structure like this, or the sequence like this in the RNA. You can see this yellow marked sequences in the RNA are complementary to each other. You can cross check that. There is 2 guanine at the 5' end, and there are 2 cytosines at the 3' end. Like that, they are complementary. So once they come like this, they can pair each other. So this structure is what we call the stem loop structure or hairpin structure. So this formation is important for row independent termination process in bacteria. So this structure will give you an idea. So there is the transcription machinery, that means polymerase enzyme is there. You can see the newly synthesized RNA molecule in green color, and there is the hairpin formation, and there is the long or the adenine sequences or the uracil sequences in the RNA, which is bound over the adenine sequences in the template DNA. And one important thing you have to notice, an AU bond is a comparatively very weak bond. So this weak bonding will cause a kind of termination. So how the termination occurs, it's a concept that you can see the first image. So it's a thread-like molecule. So once the thread-like molecule in the duplex or in the system is forming a hairpin-like structure, that creates a pressure in, and this pressure could not be this pressure causes the release of the RNA molecule from the duplex, right? So that is the third one. This interaction or the weak interaction of the AU bond facilitate the dissociation of the newly synthesized RNA. So it is simply like there is the DNA molecule, right? There is the DNA molecule, and here you can see the enzyme complex, and now suppose this is the newly synthesized RNA molecule, and that forms a stem loop structure here, and this stem loop and the rest of the molecule is here. So this stem loop formation actually causes a pressure in these directions. So once this molecule is being pulled down, the AU bonding here will be lost. So this pressure formed by this stem loop formation causes actually the dissociation of the molecule, right? So that's about the row independent mechanism. Now the row dependent process, in which there is a row protein involved. Row protein is a unique protein with an ATPase activity and a helicase activity, and which is having which can interact with single stranded molecules like RNA, and they interact usually with a specific sequence called RUT sequence, and this is nothing but the row utilization sequence or row utilization site in the RNA molecule, right? And this molecule, it's a hexameric molecule with ATPase and helicase activity, and this is the fashion how they are bounding over the newly synthesized RNA transcript. So the thing is like the hexameric molecule, the raw protein will interact with the RUD site, the raw utilization site, which is actually rich in cytosine residues, and this site usually resides at around 100 nucleotides away from the another sequence called row sensitive posi. It's nothing but a sequence like in the row independent process there will be some AT rich region, and once the raw protein is bound over the RUD site with the help of ATPase activity, they use the energy from ATP and they will move along the RNA polymerase, and these polymerase will reach in the duplex region, that means where there is a DNA RNA hybrid structure, and once they reach the hybrid region, and since they are having the helicase capacity, with the help of the helicase activity what they do is they unwound the duplex, that means here the duplex is the DNA RNA hybrid, so the duplex will unwound, which causes a release of the RNA molecule from the duplex. So this causes termination in case of row dependent mechanism. So it's an energy consuming process with the help of an external protein called raw protein. Fine. So by that we will wind up this part of discussion about prokaryotic transcription. So this slide shows you an outline about the prokaryotic transcription. The reactions number from 1 to 3, the 3 reactions are part of the initiation, where you can see the RNA polymerase enzyme and the sigma factor binding over the promoter region in the gene, and from the 4th onwards you can see the elongation process. The sigma is released out, then there is a elongation process going on with the synthesis of the newly synthesized RNA transcript, and from in the 6th and 7th steps you can see the termination. Here it is shown, it is the row independent mechanism. So by that the RNA molecule will be released into the cytoplasm, and where they are undergoing a minor processing like polyadenylation, and thus you normally as already we discussed in the previous video that this process of transcription is usually coupled with translation. So normally even once a short stretch of the RNA is synthesized, the ribosome machinery will catch up the RNA molecule and they will begin to synthesize the proteins from the RNA molecule, right? So meanwhile the transcription occurs, the translation also would have occurred in bacterial system. Fine. So that's about this part, and I hope you understood some idea about the prokaryotic transcription, and thank you for watching.